Good morning, afternoon, and evening to the more than 3,000 participants from 88 countries for attending the conference. Welcome to this morning's Funders Forum. The Earth Biogenome Project is now well on its way to achieving its phase 1 goal of sequencing and annotating the genomes of 9,300 representative species from all of the eukaryotic families. This progress has been largely enabled by funding from a number of sources, mostly governmental agencies, the private sector, and philanthropy. The project seeks about $400 million to complete phase 1, of which about half has been already raised. And completing the project over a 10-year time frame will require about $4 billion. Obviously, this is going to be a major challenge given the global economic problems caused by COVID-19 and the environmental catastrophes that have been caused by climate change. Today's Funders Forum will celebrate investments made to deliver the phase 1 goals of the project, and will explore the motivations behind providing the financial resources to the project from different funding agencies. Our guest panel will also explore the question of how we can build momentum to secure funding for the growth and completion of the project. The moderator of today's panel is Dr. Julia Wilson, who is Associate Director of the Wellcome Sanger Institute. Good morning, Julia. Good afternoon, Julia. And Julia, in her current position at Sanger, builds support for relationships that are needed to support the strategic vision of the Sanger Institute. And these interactions range from interactions with the academic, scientific community, commercial partners, science funders, and government. Julia has been intimately involved with the Earth Biogenome Project since its beginnings. She worked with me very closely to plan the official launch into November 2018 at the Wellcome Trust in London, and has been just an excellent partner throughout the past few years in all activities and linkages to the Sanger Institute relating to the Darwin Tree of Life Project and the Earth Biogenome Project. So Julia, the floor is yours.
Se me quedó mi tarjeta y...
Thank you, Harris. It's my pleasure to introduce the panelists briefly before we start the questions. So on our panel this afternoon, we have Michael Dunn, who's Head of Science at Wellcome Trust London. We've got Arturo Luna Tapia from the Colombian Ministry of Science in Colombia. We've got Andrew Gilbert from Bioplatforms Australia. Zun Zhu, who's representing BGI in China. Stephen Kappers from the US Department of Agriculture, and Sarah Bender from the Gordon and Betty Moore Foundation in the USA. And all of these organizations have directly funded and contributed to the Earth Biogenome Project goals. So I'm going to ask the panelists various questions, and then we'll open the floor to questions from the audience. And so I'm going to start by asking some of the organizations what inspired your organization to invest in biodiversity genomics. And I'd like to hand that initially to Michael Dunn, please, at the Wellcome in London.
Thanks, Julia, and hi to everybody that's listening in. So for those of you who don't know, Wellcome Trust is a global charitable foundation. And very broadly, our mission is to improve health through supporting and funding research. As an organization, we've been around for about 85 years, so quite a long time. And in that time, we've evolved over that time, both in terms of our size and our interests. Historically, we've been interested very much in infectious disease biology, and that kind of goes all the way back to our founder, Henry Wellcome's floating laboratory on the River Nile. And maybe more recently, and maybe what we've been more known for in recent history is our involvement in the Human Genome Project. And I think it's fair to say that the Human Genome Project and our support for the Sanger Institute's role in that, and subsequent investments in trying to understand the consequence of genome variation and the function of the genome, I think it's fair to say that that's all been quite transformative for biology. But I think it's also been really transformative for Wellcome as an organization and how we view the world. The importance of partnerships, the importance of ensuring equitable and open access to resources, and of course the importance of the power of big data in areas where we think that Wellcome can play an important role. I think important also for Wellcome in this is the ability to influence policy and advocate for the science, and advocate for science to be done responsibly. And that goes alongside clearly the importance of the questions that we're trying to address for science and health. And then, of course, that broader engagement, which we care about deeply with publics who should be ultimately the beneficiaries of the work. So when the idea of sequencing all of biodiversity was posed to us in the organization, I think it's an understatement to say that we were intrigued. I mean, the sheer scale of the ambition, the technical challenges, the volumes of data were really beyond anything we could imagine. And it was quite clear to us that this is the sort of project that will require big, bold thinking, and requires really active engagement from funders to think about the problems with the scientific community and try to figure out a way of how it can all come together. For Wellcome, which is actually quite a biomedically focused funder, I still think that in a way the scientific vision, although some may have questioned why we were doing it because it's not so related to health, I think quite clearly for the organization, we felt that actually the scientific vision for the Earth biogenome is really a no-brainer. We know so little about life on this planet, and we also know that so many fundamental discoveries in biology have been made through studying organisms that are not humans. I think also probably fair to say that in the last few years, Wellcome has become much more increasingly interested in the heating Earth and the impact that that has on health. So in a way, there's a kind of a real congruence here in terms of the wider impact of climate change to health, and also our understanding of basic biology and all of the wondrous things that will come from that really resonated with us, and that wider impact that we want to have as an organization. So I think probably in summary, the Earth Biogenome Project, it's a really big, bold problem. And I think Wellcome, we like to see ourselves as kind of a big, bold funder, quite willing to tackle some of these big problems and work with the scientific community to try to address them.
Thank you, Michael. That was really clear. I wonder if I could ask Sarah Bender from the Moore Foundation the same question. What inspired your organisation to invest in biodiversity genomics like this?
I'd be happy to answer. So for a bit of brief background on the Moore Foundation, we're a philanthropic institution. We were founded in 2000 by Gordon and Betty Moore. We're situated in the Bay Area in California in the US. And our goal is to create positive outcomes for future generations. So in pursuit of that vision, one of the goals of the foundation is to foster path-breaking scientific discovery within our science program. So this is one of 4 programs at the foundation. The other 3 are environmental conservation, patient care, and then a program focused on the Bay Area in California. I sit on our Symbiosis in Aquatic Systems initiative, which is housed in our science program. And this is basically a decade-long initiative that's focused on generating a fundamental understanding of how symbiotic associations function and participate in ecology and evolution in both marine and freshwater systems. So similar to the point that Michael made, we're really interested in supporting fundamental scientific discovery. So the first grant that we made in this initiative was to the Wellcome Sanger Institute, and it was to support the development of what we're calling fundamental research infrastructure. So in this case, to support the sequencing of 1,000 genomes, and that's where our connection to the Earth Biogenome Project comes in. We have this project going on for the next 4 years now. So you're going to hear some overlaps, I think, in our answers, but we viewed the support as a necessary launch point for asking questions oriented around, for us, the mechanisms of symbiosis, to help researchers establish new models for studying symbioses and more broadly other questions in the life sciences. And then very important, as important as the genomes, is to bring the community together around this shared resource. The possibility of also having conservation outcomes, while that's outside of our immediate interest with our initiative, is seen as an additional benefit. And then in working with the Wellcome Sanger within the Tree of Life program, we were really excited by the potential to elevate the impact, not only for our community of grantees, but also for the broader community of science.
Brilliant. Thank you, Sarah. I was going to ask Zhengzhu if you could briefly state why BGI are interested, and then I'm going to ask Stephen the same question.
Yeah, so a little bit introduction about BGI. BGI was actually founded in 1999. At that moment, BGI, the only purpose to found BGI is to finish the first human genome project. So the BGI's gene is actually for international collaboration. So we strongly believe that the genome technology will benefit all the human beings. So it's not only for the human's health, it's also the genome technology will also benefits all the biodiversity. We strongly believe that the technology in genome can help to protect the biodiversity and protect the environment, and can able to use the biodiversity as a resource for the new economic. So, so that's why for BGI, we actually not only doing the human genome sequencing. At the beginning, BGI was already involved in the rice genome, panda genome, and all the other vertebrate plants genomes. And we really invest a lot, and we still continue our commitment that BGI will invest in the basic research for the Earth's Earth genomes, for the Earth's life genomes and biodiversity.
Thank you. Stephen Kappers at USDA, can we ask you the same question? You know, why would USDA invest in biodiversity genomics?
Appreciate you and Dr. Lewin inviting ARS to participate in this forum. So ARS is the internal research arm of USDA. So all of our funding goes towards about 640 projects, so it's pretty well directed towards that. And our mission is to deliver scientific information and solutions to national and global agriculture challenges. So per se, we don't have a mission for biodiversity genomics, but we invest a lot into sequencing of genomes of ag species, plant, animal, microbe, pests, even beneficial insects like bees and insects for biological control. And we sequence a lot of those related species. So the Biogenome Project fits very well with what we do relative to agriculture. And so we're looking at not only a diversity across species, but even more importantly, the diversity within species. We've got a related project, the 100 Ag Pest, where we're sequencing 100 agriculture pests, and we've made a lot of progress in the last 2 years. Us and some other partners have gone from 6 arthropod species being sequenced and assembled, to now that we're at 32, representing 12 different families using the EBG genome assembly standards. So we're excited to be part of this project, and we know we're going to gain a lot more from the project than what we contribute. So thank you.
That's great, Stephen. Thank you. And I wonder if now I could move to Andrew Gilbert at Bioplatforms Australia. And obviously, I'm interested to know your motivation for being involved, but also, you know, as well as funding, Andrew, what other attributes could your type of organisation bring to this consortium? So initially, what your interest in biodiversity genomics, and then what additional added value you can bring.
Thank you, Julia. Bioplatforms is an Australian national effort around building resources, infrastructural resources really, for genomicists and life science more broadly. And we came to the conclusion a long time ago now that not only were laboratories needed, but actually open and accessible data resources. The laboratories really only exist in many ways to ensure people have the information that they can turn into knowledge further downstream. And so we made the decision some time ago really to cut the corner, if you like, and start preparing large-scale data resources in support of Australian-centric, and I apologise somewhat for that, given this is a global audience, but Australian-centric scientific challenges. And to that end, we went about a reasonably systematic approach of preparing these resources, for instance, continental and oceanographic microbiome efforts, working very closely with our museums and herbaria in preparing data sets for our very unique fauna here. Think mammals and reptiles and in time birds, but we also have very significant programs around our plant life. It's an enormous undertaking here in Australia. We have 80,000 beetles alone, for instance, in one small subset of our biodiversity. And so we've really embarked on what Harris has actually fostered globally, but for Australian purposes for some time now. The very nature of our programs, and I think it lends itself well to this global agenda, is that our efforts are open and accessible. Not just the data, but actually the specimens in behind them, the know-how and expertise that goes with that, workflows, both the chemistries, but also the bioinformatic. All of these things are 100% available. Just go to our website and you can go through and find them. And so we're really keen to ensure, and just as Stephen said, reasonably keen to ensure whatever we could produce was more broadly useful for use and reuse outside of our own borders. But we knew by doing so that our research fraternity here in Australia would gain much through collaboration and gain much through access to others' resources and know-hows around the world.
Brilliant, thank you. And Arturo, I'll ask you the same question. Just the Colombian government interest and investment in Earth biogenome related projects. What was the motivation behind that?
Hi, Julia. Good morning, good evening. Well, my name is Arturo Luna. I'm the manager of the National Biotechnology Program. I'm also head of the Colombia Bio Program. We are working now in putting together a mission on bioeconomy, and one of the main aspects of this mission is genomics. But not only genomics, but also the complementation with other omics and data science, artificial intelligence, for example. And what we want is to actually make a better use of our biodiversity. You know, Colombia is really highly biodiverse country, but we think we are not using that biodiversity wisely. And I think genomics and all the information we can get from the applications of the technologies can lead us to a better use of that biodiversity. We want to accelerate the processes of generating new bioproducts and also some new services, making better decisions regarding conservation, and also the implementation of a new protection areas around the country. I think decision making based on information is a better way to go in Colombia. You know, we have like thousands of species of birds. And one example we are working on right now is to study how the populations of birds have changed over the last century based on some previous expeditions performed by the Natural History Museum at Korea University 1 century ago. In cooperation with the Erlheim Institute, for example, what we want to do is to see how the population has changed and how we can manage those populations remaining, and also how that information can lead to new opportunities for the people in the regions where the biodiversity is a potential source of economic income without destroying it. I think the best way to preserve our biodiversity is to give opportunities to the people to use it. And genomics is one option to go.
That's great, thank you. And obviously, Harris and the team have been trying to build a global community. So I'm going to ask, particularly maybe ask Andrew first. Would your organisation, see benefit in joining a global consortium to both fund and meet the wider goals of the Earth Biogenome Project? Or should we take a sort of jigsaw puzzle to the world with national funders? How do you see that?
I actually think it needs to be both, Julia. The essence of national funding, not just within Australia, but elsewhere in the world, is that, of course, we will focus largely, I think, on the research agendas of our local research fraternity and ensure that they have the resources and genomic outputs in this instance that they need. But there's no reason that can't be linked to a more global effort. I think it's really important, though, to ensure the context within which those resources are built are potentially co-developed across the world with like countries, with like agendas, to ensure, for instance, we have suitable phylogenomic characteristics that are systematically addressed. We look across taxonomic trees in a rational way to ensure that referential information is efficiently produced and lendable across borders and so on. And so I think actually it's not one or the other. And we can't possibly meet the scale that Lewin is looking at delivering without, I think, somewhat of that jigsaw or patchwork approach that you referred to.
Yeah. Okay. I wonder if I can direct the same question to Stephen Capes at the USDA. Could your organisation benefit from joining a global community, or are you restricted to funding in your jurisdictional area?
Earlier, we're an intramural research agency, so our funding is tied to specific labs and specific projects. So we unfortunately can't make funding available, but we definitely want to contribute in any other way that we can. We definitely look at collaborations. We can host scientists in our labs, and we are and will continue to be an open source for providing not only genomic sequence and assembled genomes, but also germplasm. We've got a really strong plant and animal germplasm program. We've got the GRIN Global. We've got crop and animal genetic and genomic databases. So we've got lots of resources that we can partner with others, and we make our information publicly available. So yes, we want to be a strong partner. Thank you.
Thank you. And Zhenzu, I was wondering, we've started to talk about, funding's important, but what other attributes could BGI bring to the wider Earth Biogenome Project? I'm thinking collections or collaborations.
Yes, I think the Earth Biogenome is really an important grant project framework that under such grant project that there's a lot of collaborators. So I think that one of the reasons why BGI want to be part of Earth Biogenome Project, because we were able to collaborate with global scientists in this field. I think, and we also trying to contribute on this framework. So right now we are working on 3 major projects under Earth Biogenome, including like 10kp 10,000 plant genomes, B10k burst 10,000 genomes, VGP vertebrate genome project, and we already finished almost like 1,680 species, including like 880 animals, 300 plants, and 500 marine genomes, and about 30% of them already made public available. And all these fundings, we spend more than like 12 million already US dollars in these genomes. most of this fund is actually from BGI ourselves, kind of like a sequencing in-kind support for this project. But you know, with BGI alone, ourselves is not experts in all these fields. So that's why we have to find collaborators and find scientists in different fields in this Earth biogenome. So I think that on this framework is really important for us to expand our capacity from one species to all the other species in different diversity genomes.
Thanks, Sanju. And Sarah Bender at the Moore Foundation, I was just going to ask your views on, you heard Harris outline the significant cost it will take to deliver all the goals of the Earth Biogenome Project. So how can we build momentum with funders to secure this longer term ambitious funding for the growth of the project and the delivery of the project? Sorry, you're on mute still.
There we go.
Yeah.
I was looking for the button. Yeah, this is a great question. I think I was reflecting on this as others were speaking, and I think in my position at the Moore Foundation is to get outside of the kind of, well, now it's the virtual walls of the organization, and to learn what folks are working on and what excites the community, what's of interest, where the demand is. And it's also very helpful when we hear from the community what's going on. So it's been wonderful to see all the effort and excitement that's come out of this week-long workshop, where scientists are highlighting the work that's being done, showing kind of a point in time where we are and what's possible, and kind of delivering that in different milestones and pieces so that funders have a sense of what's needed. So of course, there's the financial investment and seeing what that looks like in chunks, if you will, right? There's the big vision, but then how can we kind of slice off so that the whole, the parts eventually lead to the whole. And then also thinking about, as I mentioned, resources beyond just financial. So how other funders may be able to engage if finances aren't available, but there are networks to leverage. There are ways of bringing folks together on platforms to build community, to share, even something as simple as just letting others know about the opportunities. And so in the case of us kind of finding our way to the Earth Biogenome Project and the Wellcome Sanger, conversations with Mark Blackster at the Wellcome Sanger Institute, it took several conversations for us to figure out where the synergies were, because the Moore Foundation came in with our interest in aquatic symbiosis and our goals, and then Mark and the Sanger team and the broader EBP had their goals. And so also just having kind of the patience to continue the conversations and check in, and there's often alignment. It just takes some time to kind of figure out where that is.
That's a great answer. Thank you, Sarah. Can I also ask the same question to Arturo Luna about, how do you feel we can build this momentum to secure the long-term ambitions of the Earth Biogenome Project?
I think through cooperation, if we all start working together from all of our countries, not just, for example, Colombia, we can keep this momentum going for a long time. And also trying to convince people, or every citizen, about the importance of this. Something I once said is that we need to bring terms like bioeconomy to the table of every home in our countries, so people understand why that matters, and also in the long term that can help to make better political decisions regarding the use of biodiversity. Also through cooperation with international partners, that's something we are looking for from the Ministry of Science, Technology, and Innovation, because in countries like ours, even though we have some capacities that can be applied to projects like VP in genomics and high performance computing, we still need some technical support from other countries. And through cooperation, we don't need just money. Money is not like the bigger issue. It's just the technical support can help us advance quite quickly.
Brilliant. Michael Dunn, I wonder if I could ask you the same question before I go to the audience questions. You know, you were welcome were an early adopter, an early partner in the Earth Biogenome Goals. How would you go about encouraging others to join us?
Well, I mean, I guess it sort of relates to one of the previous questions about the national versus international, and I think having very, very clear goals for the overarching consortium really helps individual funders establish where they might be able to fit. I think having, being able to have kind of regular open dialogue between the funders, between the funding agencies that are interested as well as the scientists is important. So I think having structures to enable that, because I think, funders kind of do like to sort of understand how their own investments might sort of be either synergistic or leverage funding from other agencies. So that's kind of important.
Mm-hmm.
And I think, kind of like picking up on some of the points about sort of, how do you build capacity? I think that's something that as a group of funding agencies, with something like the Earth Biogenome Project, that we ought to be thinking about how do we enable participation from in all kinds of settings globally, and I think that's really important. So I think that kind of the wrapper around all of that, I think, is just very kind of clear governance. So kind of what are the rules of engagement, and what does it mean to be part of the project? What are the responsibilities, and what are the I guess just by joining the consortium, what's expected of you and what do you hope to achieve?
Mm. So do you think it's something that we can convene to bring funders together and update you, and then allow you to formulate your own plans as well? But we've got that convening power to bring funders together potentially.
I think that would be valuable. Yeah.
Well, I've got my screen up so I can see all the questions coming in from the audience. I wonder if we should dip to questions from the audience now, and then Harris and I have more questions should we need to, but they're coming in thick and fast from the audience. So, and the audience are voting them up as well. So I'll ask the most popular questions first. So what do we have? Gosh, so this is to the fund. This will be directed more to the funding agencies. Are you more likely to fund diverse pan-species projects or more branch geographic specific projects? And that's from Emma Teeling, and she's talking about BAT1K. So are there funding organizations with a special interest in certain species, or is there more opportunity to fund more pan-species applications? And I'll probably direct that to Sarah first. Then I think Andrew's already answered that, so I'll direct that to Sarah, and then we'll come back in.
Yeah, sure. And so for at the Moore Foundation and in our science program, we don't have a geographic focus area. So in that case, it's really, again, kind of looking at our goals and thinking about species broadly. The project that we have going on right now is aquatic symbioses, and that is global.
That's a great answer. Yes. Would anybody else like to answer that one, or should I zip through the questions?
Well, I mean, I think it kind of relates to the question about the overarching package that we need to kind of have in place to ensure that the whole project gets delivered. And then that enables funders to kind of pick and choose the things, because some funders will be quite constrained and some won't. So I think, having that sort of sense that the funding agencies can then sort of decide where they can best have an impact and leverage. And, funding agencies like to have their names associated with certain things. That kind of mixed model then I think allows for the diversity of funders that you'll want and you will need to make this happen.
Yeah, that's great, Michael, thank you. And we've also got a question from Eric Jarvis, who leads the Vertebrate Genome Project. And his observation is that many funders seem interested in supporting the science that that's generated from genomes, but there's fewer funders who are interested in supporting generating the high quality genomes. And you know, that's the basis of our project. So you know, as funders, can you invest in generating those high quality genomes and then the science on the back of them? Sorry, and I will focus on Sarah, Michael, and then potentially Andrew there again.
Yeah, I'd like to think we're one of the funders focused on the high quality genomes, and we recognize the utility of those for kind of the launch point to all these questions. We are definitely working to share that messaging with the funders that we speak with. There's an organization, a global organization called the Science Philanthropy Alliance that is focused on getting more dollars invested in fundamental research for to target your very question, Eric. So we're trying. I think it never hurts to have repeat messaging from the scientific community of what the need is and why. And this point has come up a few times, but just show what types of questions you can answer when you have these data sets and how broad reaching they are, so that you'll kind of pique the interest of a conservation funder here, maybe an agricultural funder there, and everyone in between.
Yeah, I mean, this is the perennial problem. I'm afraid that, you know, a lot of funding agencies really like the cool biological question, but are less willing to put their hands in their pocket to generate the data that actually you need to be able to answer the question in the first place. But it's hugely important, and I think, you know, it's up to all of the funding agencies and up to the scientific community to help make those arguments. I know that a number of years ago, the European Bioinformatics Institute did a piece of work that kind of showed how much these fundamental data sets actually brought back in terms of GDP and the kind of economic benefits to society, and I think these are the sorts of things we kind of really need to just kind of like hammer home. It's a real win-win.
Yeah, and can I pass over to you, Andrew, there as well, because obviously in Australia you have managed to set up that infrastructure to enable the scientists, that genomics infrastructure to enable the scientists to generate these high quality genomes. And you know, what's the ethos there, and what pitfalls and hurdles did you have to overcome to set up that infrastructure?
Yes, we almost have the reverse answer to some of the other parties on the panel, but that's our job as an infrastructure provider to in fact prepare these data sets. And in some ways, it's funny, we don't like to prepare the data sets without the research funding to ensure the immediate utility of the outputs. Otherwise, we don't want to be in the business of stamp collecting. We want to be in the business of enabling high quality science and novel applications and so on and so forth. But we also like to prepare genomes understanding that they are a reasonably permanent resource, that they're useful for the question at hand today, but they'll be mined and used and reused by parties we might not even know of, asking questions we don't even understand today at some time in the future. And so in many ways, we think it's really, really valuable preparing genomes. We do more than genomics, but we're preparing genomes. But what comes with that is ensuring they're of a suitable quality and described well enough such that third parties can then use them meaningfully.
That's brilliant, thank you. I want to build on Michael's point actually about, you know, about the value of the genomes produced, and as you said, Andrew, we don't know the scientific question a lot of the time. So what other key metrics can we use to show the inherent value of the genomes produced? And I wonder if I can direct that one to Steve and then Zhangzhu, just what added value do you see in these genomes, and what metrics should we be using to describe the success of the EBP? Stephen, can I ask you that one?
Sure. Thank you. Sorry. So we see a lot of value in sequencing a very broad range of all the species. We do a bit of work on now sequencing wild relatives, and we're getting a lot of information out of that. But we know that that's just a small portion of the knowledge that we can gain, and in our case, to improve agriculture. I was thinking, kind of reminiscing, Harris, when we were working on getting money to sequence the cow genome, and we worked with our international partners. And a number of years after we had drafted the first version of the sequence and assembled it, I was asked to give a talk to our cattle producers here in the United States. And I started out with that. One thing I learned from that project was that I don't have any capability of predicting the future, and that I woefully underpredicted the value of having that genome sequence. And to me, this is orders of magnitude above that. So yeah, we can't even predict how valuable it'll be. So I think we need to come up with the use cases that we have been successful with showing the value of this information. And I think it's the funding organizations, but I think we need to go to our stakeholders too, and we need to make sure they're informed of the value of doing this.
Brilliant, thank you. Yeah, Zhenzu, can I ask you that similar question? What are the additional metrics that BGI would be looking for to see value in these genomes, or to help us sort of prioritize EBP funding requests?
Yeah, before, when we can only sequencing one or few genomes, so we have to pick out the most valuable and the one we have stories. But right now, since sequencing genome is much easier and the cost is now bring down, and we strongly believe that all the genome has its value. So from biodiversity and from the evolutionary important positions, that every genome has its own positions in the tree of life. So I think when you're sequencing a batch of several nodes, important species from several nodes, the comparison between the species, so the genomes of the species already bring a lot of value to understand evolution, adaptation to the environment, and the evolutionary history of this species, special branch of the tree of life. So I think it's not only the value of these genomes, only is the value from the comparison with the others, with all the other genomes. So I think currently, since the cost is bring down and the genome is much easier to be finished, all the genome is now valuable. But we trying to right now, when we do, for example, in the 10,000 genome project, we trying to, firstly to finish those genomes in the families, in the family which is not being sequenced. So trying to, fill the gaps of the tree of life. And then we trying to sequencing the important nodes of the evolutionary, the tree of life, trying to find the important evolutionary nodes of the species. And then we were also thinking about if the genome is kind of interesting with a lot of stories of the adaptation and to the environment.
Thank you. And our most popular question on the panel currently on the website is, if I want to start an EBP branch in my country and write a letter to my funding agency, what's the first question that will attract their attention? And I'm going to direct that to you, Arturo Luna, because you're representing a nation there. You're muted, Arturo. I wonder then if I can, Arturo. No. Andrew, could I direct that one at you instead? So what's that punchline to start a EBP branch in a country? What would be that headline message you would want to receive?
Look, I can only talk from our perspective, but Australia's a funny country. We're rich and highly removed geographically from the rest of the world, and so actually that connectivity, the collaborative base that the EBP brings, we produce something like 4% of the globe's science. That means there's 96% of the science that we don't have, right? And so joining initiatives such as this really allows us to leverage our unique position in the case of biodiversity, but to access a breadth of scientists, their knowledge, their know-how, and their outputs in a way that we just can't do. We can't possibly tackle this by ourselves.
Thank you. Thank you. Arturo, would you like to answer the same question?
Yeah, I'm sorry, I apologize. I had some problem with the microphone. Something that should be attractive for us is practical solutions to a problem. For example, we have some issues regarding agriculture. If you offer us some solutions in agriculture based on genomics and other omics, that would be really attractive for us. The other thing is because we need to prioritize in the funding. If that solution has multiple applications, it's even better. Even actually, we are doing some prioritization of some species in Colombia before the launching of one program we call Colombia Genomica, because we need to optimize the resources we have and also get the better use of that investment. I think at least for us, it's a really good way. Just practical solution, simple.
Yeah. Yeah, I think we've got quite a few people in the audience who are wanting to create their countrywide EBB projects, so this guidance would be really helpful. I've got a question, are any of the funders engaging with private funds? And is there sort of philanthropic funding or private funding that the FBIA Genome Project should be going for as well to help us think bigger? So Zunzu, can I direct that one at you? Are you engaging in private funding?
Currently, we are not engaged in any private funding. Most of funding for our work for this EarthBio Genome projects come from our self-funding. So as PGI, as a private company, so we fund ourselves for this project. We think this is important. We have like about 20% is come from the grants, the project-based grants from government. But they're not for the whole project, for species to species. Depends on different situations. I think currently trying to raising private fundings in China for scientific research is still not that popular. I think we in China is not quite feasible right now.
Okay. Would anyone else like to volunteer if they're working with private funding, or?
I think so. I mean, we are not doing it at the moment, but I think the answer has to be we need to. I think we need to have private agencies, national governments, commercial companies. I think this is a coalition of people that have the vision and the foresight to understand the importance of this endeavor.
I can join you also with an answer. We are designing now a mission on bioeconomy for the country. And some key aspect is the participation of the private sector and some private institutions. We need to put to work all the society. That includes the private sector. I think something that the private sector has is when they make investments, is that they have some specific goal in the medium, the long term, and that can also help us to optimize the resources. Something we want to with the Colombia Genomica program is that even though it's not implemented, we hope to launch the program next year, is that the participation of the private sector can help us also motivate some other participants to invest in this program, and also to find solutions that actually can have some applications in the private sector, so can also help boost the economy. And also, of course, the participation of the private funding is going to be key. Obviously, we don't want the private sector to do most of the investment, but at least reach to 30 or 40% of the program in private investment.
Right. Andrew, I noticed you were unmuted as well. Did you want to add in about private funding?
Yes, I think there's huge opportunities ranging from philanthropic contributions, and we certainly partner locally here with such organisations. But we found that, for instance, a number of the big miners in Australia, BHP and Rio Tinto, are really interested in partnering in these programs. They're really aligned with their social good arms, really aligned with their restoration needs, come post-economic value from mining, for instance. As an adjunct to that, and I think some of you would have seen Caroline Hogg's great talk yesterday on our threatened species initiative. There's real opportunity with partnering with commercial organisations who have mutual value, and Caroline works very close with Amazon, for instance, to ensure that data and tools are as broadly available as possible, and Amazon has been a very significant pro bono partner in that program of work, and so there's lots of ways to creatively involve people for mutual benefit, I think.
Thank you. We've got a question from Pringe, which is a good one. He said that often the Earth Biogenome Project is seen as beyond the constrained mission of a funding agency. So I'm going to direct this one to Sarah. How did you get your organisation to think big and fund these ambitious goals?
We were excited by how organized the EBP was. And as I mentioned earlier, it took several conversations with folks at the Wellcome Sanger to find alignment, but it was very clear from the get-go that we would be able to move towards achieving our goals and also fit into the goals of this broader program. So it took conversations, finding alignment, communication, and really hats off to the organization of this program for being very clear on the near-term, long-term goals, who's involved, and being transparent about those processes.
Michael, can I put that one to you as well? How did you convince your organization to think big?
Yeah, well, I mean, I guess Wellcome likes to think of itself as thinking big. I mean, we certainly, aren't afraid to sort of make sort of big, bold bets on things. I think maybe I alluded to it at the beginning. It wasn't totally obvious to many people within Wellcome because as a health-focused organization, biodiversity doesn't necessarily it doesn't necessarily flow from kind of many of the things that we've kind of supported in the past. But I think where we really kind of find alignment was in the kind of the multiple goals of the project. So it's not just scientific, it's about building a kind of a community. It's about the kind of the ability to engage and enthuse the next generation of biologists across the globe. There was all sorts of different kind of social science aspects, I think, that also were very convincing to us. So I think it was that ability to have that kind of multi-pronged approach where, as I say, big, bold problems. And it did help actually that we had a Genome Institute, which yes. and the Sanger Institute, as a key partner in that was clearly an important aspect as well.
Yeah, thank you. I'm going to move now because we've got a question about technology development programs and how the $100,000 and $1,000 genome funding programs from the NHGRI were very powerful for cost reduction and that technology development as well. And if we want to deliver the ambitious goals of the Earth Biogenome Project, we're certainly going to need technology development. So how do your organizations think about technology development and to enable us? And I wonder if I could pass that to Stephen at USDA first. Would USDA focus and funding on tech dev that would ultimately benefit the wider Earth Biogenome Project?
So we don't have funding set aside for technology development, but definitely in our mission, we're constantly looking at, do we need to develop technology that will help, in this case, this particular project move along. And with our 100 Ag Pest, which is an affiliated project to this Earth Biogenome Project, we had some real struggles with trying to develop enough DNA out of a single specimen of some of these insects. They're very small. So we worked closely with PacBio and developed a miniaturized DNA input, and it's working very successfully. So just as scientists working together and developing that technology, I don't think that addresses the bigger issue, how do you dramatically reduce the cost? But I think each of us are going to be working with some unique species that we're going to have to develop things, and we need to share when we develop that technology with others, because that may be quite useful in lots of cases.
That's excellent, thank you. Senzu, can I pass that over to BGI as well? Because obviously you have very much a technology development program at BGI, and how could that help us realize the wider goals of the EBP?
Yes, I think that's why we strongly believe that technology is very important for the Earth Biogenome Project. That's why we are thinking about the Earth Biogenome Project, because at this moment the technology is feasible to able to finish this grand project. So, but still there's a lot of issues ahead of us, including, for example, there's a lot of genome species actually is very difficult to sequence. The technology currently is not able to solve this problem. And for example, the computational issues, because we have more and more genomes right now, to do the comparison between different genomes, the computational task is too heavy right now. We don't have the computer or the bioinformatic tools can handle this problem. So that's why we continue to invest in the new technology to solve these difficult genomes and to solve this bioinformatic issues.
That's brilliant. Thank you. Thank you. I realize with the number of questions coming in we could talk for many more hours, but unfortunately we have to close in a moment. So I just want to take the last minute to say thank you so much to this fantastic panel for engaging and being so honest with us from a funder perspective. And also offering us some real wisdom that we can build on and help realize the goals of the Earth Biogenome Project. I'm conscious that we didn't get through all of the questions, and we will attempt to do that and get the answers out there generally. But just to this, I would like to bring this to a close and thank you all for joining me from Australia, from China, from Colombia, from the US. It's been an absolute pleasure to hold this global forum of the Earth Biogenome Project. Thank you so much.